the genetics of complex diseases. And when he says next generation, I'm not so sure that it's the generation that we're in. So we'll learn about that. And, you know, we shouldn't forget, just as Barbara McClintock and others work on the maze, that, you know, you're not only working in human diseases, but with plants as well. And I learned about that when I got a chance to reconnect with you in San Diego back in June at the Pharmacogenomics and Bioinformatics meeting. So welcome back to Ann Arbor. It's an absolute pleasure to have you here. I know you've had some good visits, and the students really raved about the lunch. Thanks, Dick.
Great. Thank you. It's great to be back. Oh yeah, that was when I was a CMB student. Yeah. Yeah. And I should clarify. So there's actually 3 courses at Cold Spring Harbor. Lincoln started one in programming. Bill Pearson started one in algorithms and theoretical things. And the one that you took is actually the one that myself and Rick Wilson, Elson Chen, and Richard Gibbs started in 1995. And it was, so there's actually 3 that are still ongoing in one form or another. But anyway, it's great to be back. Thank you for the kind introduction. I'm going to talk today about largely how we have found we can move things forward in the study of complex diseases. By that I mean non-Mendelian things, obviously, just to be clear, but also the problems we're running into that I don't think I fully appreciated when we started doing this, and how we're trying to deal with them kind of intellectually and strategically. And I will talk a little bit about next gen sequencing, although most of what I'm going to talk about is going to be Illumina sequencing, kind of, which has become kind of routine now after the initial instruments came out. So I'm going to talk about introduction of sort of briefly what we look at other than the psychiatric genetics, which is what I'm going to talk about most of the time. Talk about how this dramatic change has changed the way we do things. But then I actually want to talk about the difficulties, some examples of projects, some things I think we've learned from them, but also the difficulties that have arisen. And then I'm going to just briefly mention how we're trying to change the way we approach the genetics, talk a bit more about some of the functional things we're trying to scale up, and then talk about maybe, the next step in terms of the sequencing technology. Our lab has always been heavily vested in new technology and continues to be for a variety of reasons. One being, I think, having new technology lets us ask new questions. Another much more trivial thing is it's actually fun. We enjoy trying new things. So, as I said, I'm going to talk largely about psychiatric genetics, but really it's this larger question of the functional impact of genetic variation. And I think, as I've been talking with people, I've had some great discussions with people today, and it's, I think, clear to a lot of people here that this is how genetic variation affects a phenotype is actually a very complex question. And it has a lot of different aspects that we're just beginning to uncover. And I have to say, we're not actively doing that with most of what we're doing because we haven't figured out how yet. I think we're getting to a stage where we know it's a problem, and we're trying to figure out how to do it. But I will talk about how we're trying to simplify some of the functional aspects. And then we also look at genetics of cancer progression. I'm not going to really talk about it all. And then we have a fairly large effort in how to use new sequencing technology to sequence large plant genomes. And since this is a kind of a medical school environment, just so that people know, most plant genomes are as big or bigger than the human genome. So the wheat genome, for instance, is a 16 gigabase hexaploid genome. And to sequence that with 100 base Illumina reads is not going to happen in a straightforward way. And so we're trying to figure out new ways to do that. And we've been involved in most of the large plant genome projects over the years, Arabidopsis, rice, maize, and we're now trying, and those are all small genomes, except for maize, it's about the size of human. But rice and Arabidopsis are small. Now we're trying to really export that. I'm not going to talk about that. but it's an exciting area. You've all seen this, but it is from NHGRI, and it's basically the cost of sequencing a human genome. And it's plotted next to Moore's law. Gordon Moore, one of the founders of Intel, on how many chips you can put on a surface in a given size and how that changes over time. And you can see when the Illumina machine came out, we were a test site for Solexa. We got our first machine right at the beginning, literally between Christmas and New Year's of 2007. 2006, and we're running it early 2007. You see when that came online, this cost dropped. It got a bit stagnant here, but it's dropping again, and it's down approaching the $1,000 range with the new X10 sequencers that are coming out. Or it seems to be, those aren't really completely online yet. Another way of looking at that is the sequence output from our lab over time. This is monthly sequence output starting from August of 2007. So this is when we scaled up the multiple Illumina instruments until sort of late last year. And you can see, you know, it's just a phenomenal increase. And it went from, you know, a few billion bases a month to pushing 10,000 billion bases a month. That change basically meant that we're not a small lab, we're about 20 people, but that a moderate-sized lab could produce more sequence data in a month than everyone in the world in aggregate had done 10 years in a 10-year time period 5 years before. That is a fundamental and revolutionary change in what you can do in genetics. Having said that, it's not necessarily straightforward how you apply that to obtain new information as opposed to new bases. When I was a student here, David Botstein gave a talk in the early days of chemistry, sequencing, and he said all you get from a sequence is A, C, T, and G. That's true to a certain extent. You get a lot more of them now. You get 100,000 times as many. But getting more does help because you can make comparisons. And so it's not as bad as I might make it sound, because you know if you can look at a lot of people, you can then look for patterns, obviously. So some of the recent things we've done, other than psychiatric things, just as an example, we have been trying to approach this wheat genome as part of an international consortium. We're still working on that, largely trying to, I said it's a hexaploid. It's actually composed of 3 related wheat-like genomes. And so we actually sequenced those plus the hexaploid and then tried to derive which copies of the genome were which in wheat. And that's an ongoing effort. And then, again, we've been involved in cancer progression, this being a good model of cancer progression. There's others as well. We're doing some work in colon cancer as well, which is also a good model for progression of cancer. But what I really want to talk about is how we can use this new sequencing capability to understand complex diseases. And this is kind of a laundry list of the things you could do. And at one level, you know, various people are doing all of these with useful information coming out of it. The first thing we tried to do with this technology, because when we first got it, you know, it gave sort of a 500 million bases per 2-week run. You know, now you get 700 billion bases per 2-week run. So it's, you know, it was a lot different then. So the first thing we tried to do is we tried to resequence what we thought was a biologically interesting region from a lot of people. I had done a previous project like that about 15 years earlier in the mid-90s when I was still at NIH, where we sequenced a region we thought might be involved in Huntington's disease gene with the Huntington's disease collaborative group. I was in Craig Mentor's lab at the time. And we sequenced 50,000 bases from one person. And that was about a $2 million project. I mean, that was big science. It was Craig and Francis Collins and, you know, it was those kind of labs doing this. And then we come with this new technology, and we were sequencing, in fact, 500,000 bases So 10 times as much from 1,500 people with about the same budget, I would say. So that's sort of the expansion. This region is named, misnamed, I think fairly, disrupted in schizophrenia 1. It was originally, the name came from the fact that the original proband in the family was schizophrenic. They found a family with a very high incidence of psychiatric disorders, Doug Blackwood and David St. Clair, in the 1990s. And then Douglas and David Porteous found that, in fact, there was a translocation between chromosome 1 and 11 that segregated with the illness largely, not completely. And what you'll see is there are people, such as here, who have the translocation who aren't sick. There's a few of them. There's not many. Most people with translocation are sick. All the people with the translocation have a reduced neurological test. So, but they're not diagnosed with psychiatric illness. This is kind of typical of families. Anyways, we wanted to see. So this one family has this translocation. No one else has been observed with this translocation. So we wanted to sequence this in the region, the entire region, not just the coding regions, but the entire 500,000 bases from a large number of patients and controls. What we thought was a large number of patients and controls. This is the result. It was a very interesting control group. The Lothian birth cohort. This was 889 people who were born in 1936. In a date in June in 1947, all 11-year-olds in Scotland were given an all-day cognitive test. And then in the early 2000s, they started putting ads in newspapers. If you took part in these and would like to take part in a follow-up, come and contact us. And so they then retested these people. And they also have their NHS, National Health Service records, so they have complete medical, drugs, everything they've had. It's a great control group. And then these are patients that we looked at. We sequenced them all at high coverage with each patient was done, each sample was done with 96 overlapping long-range PCR products, which I can probably never apologize adequately to the postdoc that did the project for doing that. I mean, because she had to optimize everything. I mean, just an incredible job. Her name is Jennifer Parla. Incredible, and she's moved on. She's had a really nice job in industry. I'm happy for that. But I mean, it was really tough to design, optimize primers and that for long-range PCR. Another thing we did when we started doing this, we noticed that we had genotype on all these people, and the accuracy was very good when we compared the genotype with the sequence. But when we looked at the variants, and this is first generation Solexa chemistry, 36 base read. When we looked at detected variants, a large numbers of the singletons when we did spot testing did not validate. By large, I mean over 20%. And we were kind of at a conundrum. Well, how in the world can we have like a 99.98% accuracy on detecting a GWAS, a SNP that's been detected by an array, and have a 20% error rate on other SNPs? It's not possible. And it turns out, and we were just thinking about it wrong, basically, singleton genetic variants and sequence errors look identical here. So if you have one error per 3 million bases, if you sequence 500,000 bases from 1,500 people, you have a lot of errors. And they all look like singleton genetic variants. And so we ended up sequencing, doing capillary sequencing on everything that was at less than 1%, which is around 3,000 variants to validate them. So these are all validated by orthogonal chemistry. Which we still do, and it's actually a bottleneck, but we're still concerned about it. We found that a lot of the variants were novel in that they weren't in the, at that time, still growing, well, they're still growing, 1,000 genome and other projects. So we identified a lot of novel variants, especially the rare ones, not at all surprising. The rare ones were more likely to be novel. And again, these validated rare ones are real. And we found some profile of exonic variants and so forth. And this was, I should say, this was all published in Molecular Psychiatry. It came out online last year and in paper over the summer. People still look at paper. I wasn't even aware it wasn't out yet. When we looked at where these variants are, we found something interesting on this in the next slide. One thing we found is a few things like these. These are protein coding changes, and this one is the most interesting. I'll show more about it. I said it was probably misnamed to call it disrupted in schizophrenia. Most of the people in the family, actually, when you look at it, are bipolar or major depression, not schizophrenia. It just happened to be the first one with schizophrenia, and it kind of labeled the whole thing. This was a variant that had been detected before by Song et al. At Johns Hopkins, and I believe theirs was bipolar. In our case, we found it in a unipolar patient. This variant has not been seen in a non-psychiatric patient in, I think there's been 10,000 genomes looked at. It doesn't seem to appear in people that aren't sick. Based on that, one would predict it's highly penetrant. Went back and sequenced other members of the family that had this polymorphism, and you can see that basically there's a diagnosis for everyone that has the variant. Of course, the problem is this person who doesn't have the variant also is bipolar, and that is a problem inherent with these illnesses in that the population frequencies are so high. This may be a phenocopy. I think we can't rule out the fact that this maybe isn't real. I think we're pretty confident it's real based on the fact that we don't see that CT variant in healthy people. But this is sort of the complexity that we're dealing with that I'll keep coming back to. The other thing we found was that there was a variant, a common variant, that's an extremely rare variant, that R37W, very, very less than 1 in 1,000. This is a common variant. I forget the exact frequency, but it's pretty high. And it is associated, when we do a gene-wide burden test, with major recurrent depression. Not everyone that has this has major recurrent depression, but again, it's associated with major recurrent depression. And it's an intron. It's an intronic SNP. And I think what we're seeing here is what we're going to see in many of these cases, whether they be GWAS peaks or cases like this, which is that there are both rare and common variants operating in the same loci of the genome with differential effects. And I think that's part of the complexity that we're seeing. And if you think about that, the perturbations of that on things like GWAS are significant, as well as on studies like this. So we wanted to move past looking at single genes. And the areas we've moved into are sequencing exomes and sequencing complete genomes. Well, it was actually, before we actually heard of the Illumina sequencer, we heard of the 454 sequencer. And it was pretty clear to me from hearing about the 454 sequencer that it was a huge step up from ABI capillary instruments available at the time, but you couldn't do whole genome sequencing. It just wasn't going to happen at any scale whatsoever. That's actually probably fair to say of the first Solexa instruments. But we thought we could sequence coding regions. And so, but it was, there's PCR on one gene, how bad that was. If you think of doing 20,000 genes, it's dreadful. So in thinking about this, Greg Hannon and I started working on, are there ways in which we can capture all the exons being some canonical definition based on whatever database you're looking at, and so forth. But the vast region of coding genome, and then sequence just that, because that's only like 1 or 2% of the genome, depending on how you calculate it, 1, 2, 3. If it's in code, it's 90, but we didn't know about that then, fortunately. And so we wanted to be able to capture the coding region, because we thought we could do that even if we couldn't do the genome on a reasonable number of people. So we developed exome sequencing to do that, and it's been a mainstay in the lab since then. Although we're doing more and more whole genome, I think, but we still do a lot of exome sequencing. And I don't actually show any data, but we actually have more recently gone to targeting very small subsets of the exome, a few 50 or 100 genes, what have you. And we really see a lot of advantage to doing that. And then people are also doing RNA capturing. So it's been a very useful technique. But in trying to apply it, what we have found is we've run into a problem with the fact that we all have a lot of variation. And how does one filter out the natural, be careful of my terms here, the non-pathological variation in the context that you're looking from the rest of the variation? And I want to talk about some simulations that we've done with Rachel Karchin at Johns Hopkins to show this problem. And then some examples of kind of how we're trying to zero in on things and where we think are the limits of genetics. So this is from our first, I don't know, a couple hundred exome sequences we did in a collaboration with Jimmy Potash at Johns Hopkins at the time. He's at Iowa now, and others at Johns Hopkins. These are bipolar patients and controls, and we did exome sequencing on them. And these are the, there's coverage information. It's kind of noisy. I apologize for that. But this is basically the total number of variants. And these are the non-synonymous variants per person. And so if you look at any of us in this room, we have something approaching 3,000 genes that have a variant relative to the reference. That's why we're all different. It also means if we're trying to do statistics on these variations in a big population, we have a really high noise to signal ratio. So we wanted to see how many people do we actually have to look at with different levels of genetic heterogeneity to how many exomes we have to sequence. And so we worked with Rachel, who's in the electrical engineering department at Hopkins, and her postdoc, Yun Cheng, and did some simulations. I want to explain a bit what they are. Again, we're trying to quantitate the impact of genetic heterogeneity. And by that, I mean, in the population of people, how many genes can cause schizophrenia? We know it's not one. We had a guest speaker at Cold Spring Harbor that said he knows it's less than 20,000. I yelled at him in the middle of his talk for saying that, but it's a fair statement. So with different levels of heterogeneity, what Rachel did is basically simulated a population with known variation levels based on the exome variant server data that Debbie Nickerson and others have made available to people. So basically made a population with variants that are based on the frequency of which they're observed in the population of the exome variant server. And then essentially electronically doped in causative mutations in 1 gene, 10 genes, 50 genes, 500 genes, and then did statistical analysis and said, okay, how many people do we have to look at to see if, a good p-value? And did I go too far? Yeah. So anyways, and I should say this is single gene detection. Network detection will work better. There's probably other bioinformatic tools that will work better, but this is single gene detection. It was a little hard to simulate networks because we don't know how many networks the genes would fall in. So, and so to some extent, this is the worst case scenario. On the other hand, we're assuming with these, I'll show you, I have some others I didn't include. These assume 100% ascertainment. In other words, if there's a variant causing the disease, it will be detected by an exome sequence. In other words, it's coding. That's clearly not true. We've also done it with 50%, and the numbers get, as you might imagine, appreciably uglier. They're pretty horrific as it is. So this is to find all of the genes that would contribute at different levels of heterogeneity, assuming that they're 100% exonic and detectable, 100% ascertainment. So this is with 1 gene causing it. This is how many cohort size is numbers of patients. There's an equal number of controls. So this is just, so these are, you double these. And what you can see is with, as you would expect, with 1 gene causing it, it's really easy to find. Because everyone has the same mutation, so that really stands out. As you go to 10, you start looking at, 1,000 or so people. This is power, sorry. So this, she basically simulated this 1,000 times, and it's what percentage of the time she detected is how she's determining power. Assuming 100% penetrance and 100% ascertainment, exactly. So it's to that extent, these are best case by far. From a bioinformatics standpoint, they're probably not best case. You can do a bit better than just doing single detection. But yeah. But when you get out to what are probably realistic numbers, 200, 500, and 1,000, you see you have 10% power with 200,000 people with exomes. It's actually possible to sequence 200,000 exomes. It'll probably be done with a disease like schizophrenia, but the community is probably in the 10,000 range now with bipolar and getting there with schizophrenia. So I mean, these numbers are doable. That's to find all the genes. To find any of them, it's a bit better. So this is to find any of the genes causing it. And what you can see is with this sort of 500, you need to get 500,000. You need to get 10 to 20,000, and you'll find things. And those numbers are approachable. But and I think they're starting in type 2 diabetes. I think they're starting to hit those and so forth. So I think this is doable, but it's also sobering in that there was a time when you thought if you did 1,000, you'd find everything. And that clearly, well, we've done more than 1,000. We haven't found anything. So that's pretty clear that, well, that's not, we found some interesting things. We need to figure out what to do next, which is part of what I'm going to talk about next. So, as I said, you can do these. And we're pooling data with people, and this is ongoing. But another thing we would like to do is go faster than waiting for 100,000. So we have from the beginning been looking at families. This is again from our collaborators in Edinburgh. It is a family that has a region on chromosome 4 that has around 20 million bases that appears to it's one of 3 regions. There's minor peaks on 2 other chromosomes for bipolar and major recurrent depression. And so we did whole genome sequencing on multiple members of this family. This shows the initial version of the pedigree. I'll come back to that. But we sequenced 3 affecteds and 2 married in unaffecteds. Whole genome at high coverage to look for variants in this region on chromosome 4. And then we took advantage of the families, the family structure. And we did filtering like this. So this is how many, you get 3 million variants per genome, about a couple 100,000 of them are on chromosome 1. This is the non-repeat. And then when you say, okay, what are the things that are present in all the affecteds but not the unaffecteds, you start filtering down. And you say, oh, if it's in 1,000 genomes, we'll get rid of it. And if it's not in, if it's not in the linkage region, and we get fights over what the linkage region is exactly and so forth. But, it's got to be in an exon. And you can get down to 1. But, after we did this, and we were quite excited when we did this, even more so when I show you the gene on the next slide. But I think let me give you the punchline here is you can do this, but would it be right? So if you look at these filter steps, I mean, if you're looking at these, you probably should be thinking, that's not a good bet with depression. 10% of the population has it. It's not like something where, nobody lives past the age of 3. 10% of the people walking around have this disease. So some of the people in the 1,000 genome may have this marker. So, we can get to 1, but, and when we do, it's fun. This is the gene. This coding variant is in a gene CC2D2A. It interacts with DISC1, our favorite gene in this area. Alleles of this, not this allele, but other alleles of this cause something called Joubert syndrome, which has as a cognitive limitation as part of a cognitive disability as part of the phenotype. I was talking to Aravinda Chakrabarty, and I said, it's a really interesting gene. And he looked at me and he said, Dick, you're old enough to know that all genes are interesting. Mea culpa. So there's some other genes in this area that have non-coding variants that have, by other studies, been implicated in other illnesses in this area, including bipolar, which is really the phenotype. So we ran a program called VAST from Mark Yandel, which has been used to find some disease-causing genes, Mark Yandel at Utah, on the whole genome of these individuals. And it predicted the CC2D2A was just under the threshold of 10 to the minus 6. It was like 10 to the minus 5, something like 10 to the minus 5. It came up with these two genes. The yellow means they're in one of the linkage genes. If I hadn't been chastised, I would say both of them are interesting too. So we were kind of scratching our heads about this, and it became we became aware that there were 11 other families that had been identified by various researchers around the world who had a linkage peak on chromosome 4 for one of these illnesses. So we had actually, it took a while to get IRB approval. We actually couldn't get IRB approval to do whole genome sequencing, but we did to do targeted sequencing on 36 million bases at the tip of chromosome 4. So we did that on all we made custom capture reagents, and we sequenced that 36 million bases from about 50 people in it's actually 11 families, not 12. And we ran VAST on it. The ones in yellow are ones that were identified in more than one family as being a VAST hit. CC2D2A is not one of them. SORC-S2 is, as are some of the others. This one was also in the, this was in the original family. This was not in the original family in the coding region by VAST, but it did have variants. And honestly, we thought, well, we sequenced the other 12, they'll all have mutation in the same one. And we weren't thinking clearly. Our enthusiasm had escaped us at that time. Because if you think about a 36 million base region and the genetic heterogeneity I talked about, this is how many genes you would expect in a 36 million base region if there were this number of genes causing the illness. And so basically, 36 million bases is a big enough part of the genome that you'd expect multiple genes if there's a lot of genes causing it in the population. And that's, probably what we're seeing here, although we don't know which ones, and we don't know what the true positives are from the false positives. So where do we go with this project? Well, it turns out that there's a new generation of people from this family, and we've just sequenced another 15, I think, members of the family with and without illness from the next generation, whole genome sequence from the original Scottish family. We're currently analyzing that data, and we're still, doing additional families. But I'll come back to this because, I said we could get down to 1, but it would be wrong. We're really trying to reevaluate how we do the filtering and then what we do as a next step. The architecture we found most illustrative in terms of finding candidates from small numbers is looking at trios. By that I mean unaffected parents, affected child, and it's not an architecture without risk. There's a possibility of an undiagnosed parent and so forth, but we're still finding it useful. So this is a collaboration with Aidan Corvin, this project led by Shane McCarthy in our group at Colston Harbor. Aidan Corvin, a psychiatrist in Dublin, and basically doing exome sequencing and looking for de novo mutations in the affected child. And we actually did a relatively small number of these. We've been doing a lot of these in autism with Mike Wigler, and really excited about the results there, which have been published by Mike's group in a variety of groups. But we did it with schizophrenia and actually found de novo mutations. And when we ran through this Neurocarta database, and there's various ways you can do these ontologies, the #1 enrichment was actually autism spectrum disorder, although it was not significantly corrected. But we were intrigued by that overlap with genes causing autism. This is probably the most common de novo mutation in autism, CHD8. This is found by Jay Shenduri, Evan Eichler, and others. And it's turned up in some of the ones we've done at Colston Harbor as well. And I think some of the ones Matt States has done at Yale. Those are just nonsense mutations. There's missense mutations. This is, I'll say this one really is interesting. This is McP2, causes Rett syndrome, which is a severe autism-like disease. Again, these CCH8, AUTS2, MLL2. And so it's a small number of families we looked at. So we find this overlap with autism extremely interesting. In addition to overlapping with autism, we actually looked at functional characterizations, basically protein domains, and found that the genes were enriched for proteins that are involved in chromatin remodeling. And that we were quite, including MECP2, CCH8. And in other contexts, this is actually a leukemia gene, different alleles. So we became very interested in chromatin remodeling. Shane actually went back and looked in the literature then at other published studies using the same set of genes that we had done in collaboration with Ivan Iosifov and Chris Vakosh, Chris being a cancer biologist, Ivan a bioinformatician. And with that list of proteins that have chromatin binding domains in them, he looked at various published studies in autism and schizophrenia, find a lot of these things cropping up. This was published in Molecular Psychiatry over the summer. This was especially interesting to us. Just to summarize this particular study, a small number of trios, I mean 42 trios compared to, we've done 2,000 case control bipolar exomes. And there were a higher than expected number of de novo nonsense mutations, but also this overlap with autism and chromatin remodeling. I said that was especially interesting because there's this growing story from the neurobiology standpoint, largely but not exclusively from Rusty Gage. This is a group in Edinburgh that basically raising the issue is somatic transposition leading to some neurological illnesses. And it's clearly, it's controversial. There's been other papers from Chris Walsh indicating it may not be the case. And I think it's, you know, it's certainly up in the air. But I think it's an exciting area. And we were excited to see a potential convergence between genes which might dysregulate this process and this process potentially being involved. So we have a variety of studies going on, some of which I've talked about. Genome sequencing, exome, case control, mostly families. And through all of them, I'm beginning to come to the conclusion that, again, we can filter down to a very small number of variants, but we really do not have a signal and noise situation that will allow us to do that without losing true positives. We will lose true positives if we filter down to very small numbers of variants. So we're trying to, and I'm not going to actually talk about the experimental details of this, but basically our plan is to get down to a reasonably small number, and then in families or smaller studies, and then target that smaller number of genes with targeted sequencing in very large numbers of people. So we can now, if we sacrifice identifying individual variants, but just look for, individual combinations of variants, but just look for, you know, what percentage of patients versus what percentage of controls have variants in a given gene, we can do close to 1,000 people in 2 lanes on a high-seq with 50 or 100 genes. And that may be the kind of numbers we need. Under $10 a person. It's more expensive if we do individual samples, but we're doing some row column pooling testing. I think we'll get that down pretty low as well. So the idea is to do the kind of things I've talked about, but then zoom in with very large numbers of samples to by one of a variety of techniques, including, you know, Jay Shanduri's been doing molecular inversion probes. We've been doing capture. I think it remains to be seen sort of at what scale, what process works better. But this is doable. And there's starting to be these very large sample sets, at least into the tens of thousands available. And so we actually have an NIMH grant to look for modifiers in the DIS1 family and then target sequence those in about 20,000 people. And so that's the direction we're going with genetics. But in addition to genetics, we think we really need to look at function. And ideally, we'd like to do that in iPS cells or mice, but that's expensive. It's very expensive. And if you filter down to 1 or 2 variants, you can maybe envision doing that. But if you say, well, I don't trust my filtering if I get the 2 variants, I've got 50 variants, that becomes really fairly expensive to do in mice. So we're actually working with Chris Hamill at Cold Spring Harbor. We're very interested in trying to do a much higher throughput assay in C. elegans. So Chris and Olivia Ramos in my lab is a postdoc working on it. And so you're all familiar with C. elegans. There's a neuron in C. elegans, the PVD neuron, that runs the length of the animal. A lot of neurons in C. elegans are refractive to RNAi. The PVD neuron is not. The PVD neuron undergoes multiple levels of arborization. And this, since it's C. elegans, it's very programmed. They all look pretty much the same, unless you perturb the system. This is a micrograph from Christina, who's actually a postdoc in Chris's lab, but she worked on this for her thesis in another lab. And this is the worm with a green fluorescent. C. elegans is so nice. I mean, this worm has been engineered to express green fluorescent protein in this nerve, right? So they've got a strain that expresses in this nerve. If you add GFP, RNAi against GFP, the signal goes away. You can see the arborization there. And here's an RNAi against something that interferes with neuronal arborization. And you can see it loses the quaternary dendrite formation. So this is, as it turns out, it's one of the genes we identified in one of our genetic studies that Christina had already knocked out. And you can see when you knock out this gene, it loses these quaternary dendrites. It's a little hard to see, but it does. And she actually, when she does this screen, she actually looks at 10 animals and determines how many of them lose the thing. And then RNAi does have some off-target effects, obviously. And so once the likely candidates are identified, they go in and mutate the gene to confirm it. So this is a list of genes we gave them. About half the genes we pick from humans have a good homolog in C. elegans. Olivia, a bioinformatician in our lab, did that. And then we put this first set we did. The red ones are ones that had a phenotype that had either under or over arborization phenotype. You can have an overexpression, an over functioning as well. In Christina's published work as a graduate student, she knocked out all the genes on one chromosome, or RNAi all the genes on one chromosome in this assay. 0.4% have a phenotype. So, you know, we're a lot more than that here. So we're excited about this. What we're actually trying to do is, we have a pending grant, and we'll see if it gets funded. If not, we'll keep working on it. But knock out all 20,000 genes, pick all the ones that have a phenotype, and then sequence those in 10,000 schizophrenics and 10,000 controls, and see if we get genetic variation in humans in genes involved in this. And we are well aware that arborization is not the only phenotype that can contribute to any of these illnesses. And in fact, CHT8 has no phenotype in this assay, which I cried over, but so be it. But we still think it's worthwhile to do, basically flip things around and approach it from function. So to roughly conclude this part, and I have a little bit more, we're really we're putting together really 2 funnels. One, a pipeline of gene discovery and then validation by sequencing in larger populations with kind of a looser net here. And then the idea is to put these in C. elegans initially, and then take the ones that look promising into the more expensive, ultimately behavioral models. And that's kind of our paradigm. I hate to use that word, but I guess it's accurate here, of how we hope to move forward and deal with the signal to noise issue. I want to talk a little bit about, I added these at the last minute because I thought people would be interested. I said we're interested in technology for a variety of reasons. when we sequence a genome or a transcriptome with an Illumina machine, there's some things that work really, really well. We're very good at calling SNPs in non-repeat regions. Very good at that. Very high fidelity, very high accuracy. It's probably 1 in 10 million error rate at worst. On the other hand, indels, various structural rearrangements, which may be more or less important depending on what you're looking at, probably a lot more important in cancer, for instance. Illumina technology has some limitations with that, very clear limitations. So we have been working a lot with PacBio, and I gave a talk at the AGBT meeting that PacBio has on their website that talks about that, but this is newer. I wanted to talk about this. So this is, you've probably heard of this, MinION from Oxford Nanopore. We're part of their early access program. It is simple enough that even I have run it. I went to the lab and they let me do a sequencing run on it. It actually worked pretty well. And what this does is, here's a picture of it hooked up to, it's a USB device. And the zoom in on it. These are from their website. This is a read length plot. And, you know, Illumina reads are 100 bases, right? You can see, you know, 10,000, 20,000, so forth. Or the peak there. James Grotowski and Mike Schatz's lab, who's working with us on assembling these data, plotted this. He also then plotted it. He kept the labels the same, so it's easy to interpret, but you get a log plot. Let's spread it out. And you know, you can see there's a big peak, sort of 80,000, but we get reads out to 100,000 bases. That's a bit longer than we get with PacBio. The error rate's higher than PacBio. So we're working on hybrid assemblies, as we do with PacBio with Mike Schatz on error correction. We're also doing error correction of just Oxford versus Oxford, as we do PacBio versus PacBio. We actually haven't done PacBio versus Oxford yet. It might actually be worth thinking about. But you know, it's very new technology. We're excited about both this and PacBio for a variety of things, including these complex plant genomes, which I talked about. But also, I mean, I talk with people here about looking at the transcriptome. You get a very different view of the transcriptome with a 10 kilobase read than you do a 100 kilobase paired into Illumina read. And there's issues with this. And PacBio, the largest one being you don't get a lot of reads compared to an Illumina sequencing run. You get billions of reads on Illumina if you want it. Here you're with PacBio, you get more PacBio than this, but with either of them, you do not get a lot of reads. So there's probably going to be some upfront molecular biology enrichment techniques such that have to be employed. But we're excited about the fact that technology is still moving forward, and we're pretty optimistic that of the number of companies trying to do this, some of them are going to hit. And I wanted to show this. I showed that USB device. This was the first, one of the first computers in a lab here at the University of Michigan back in 1980 or so. This is an Osborne 1 with the incredible 5-inch CRT screen. This was a portable computer. It weighed about 30 pounds. And you know, this is 30 years of technology, right? A USB sequencer versus this. And that it's not me. That is someone probably from the business school. But you know, that's how big this thing was. Almost $2,000 in 1980. This was an expensive thing. It had 2 floppy disk drives, 102K each, and 64K of RAM, which would make a crappy phone right now. Actually, a crappy watch after yesterday's announcement. So, conclusions of the main part without the digression to the nanopore. Heterogeneity is a real problem in looking at these. It's not the only problem, but it's a real one that we're focusing on. We're trying to do a phased approach because we think it'll let us get around some of the issues in a time and dollar framework that we're roughly satisfied with. I don't think we've done well at integrating things like common variants and rare variants together, GWAS data and so forth. I think we need to get better at that. I don't mean just our lab, I mean in general. Likewise, the encode data, I think we need to take better advantage of. There was a huge amount of data and it kind of dumped on us, and we're trying to figure out how most efficiently to use it. We're excited about the functional things, initially C. elegans, but we clearly have to move into other more complex systems, and that's part of what we're doing at Cold Spring Harbor at the Stanley Institute. In our most recent iteration, we've actually included a lot more of neurobiologists and functional people to move forward. And I had to throw this in. This is an original ear of corn from Barbara McClintock showing variegation due to transposition. We may be looking at that in the brain in some of these illnesses and somatic variation. I think we're almost certainly there. So it's an interesting reiteration of some stuff from 50 years ago. A lot of people involved in this, like to thank people from my lab. Melissa runs the initial data analysis pipeline on 9 HiSeqs and 2 PacBios from her apartment in San Diego. Yeah, it's an interesting thing. And she's been in the lab since 1996. And then Sarah's working on the stuff I didn't talk about on the high throughput targeted sequencing. Others in the lab, the Edinburgh. Sarah's also doing the Oxford nanopore stuff. She heads our technology development group as well. Our collaborators in Edinburgh, David, Douglas, who've been just instrumental. Aiden at Trinity on the trios schizophrenia. Shane's a research investigator in the group, and then a number of collaborators at Cold Spring Harbor who have provided intellectual, moral support. Greg, in particular, early as we were setting up next-gen sequencing, was invaluable, and then our collaborators I've mentioned throughout, and then collaborators at Johns Hopkins and Rachel and other people contributing. And lastly, this is not cheap. We've had very generous funding from the Stanley family and the Stanley Medical Institute, as well as NIH and NIMH, and be happy to answer any questions.
The multi-gene facet of complex diseases, where you're getting all these genes coming up, and in some ways it's almost a, if I get 2 of 3 of these out of a pathway, something bad's going to happen. Is there any way of using the C. elegans approach you presented here to use pairs or triples of these to look at, maybe it's 2 or 3 things with CHD8, and it could be, or maybe it's any one other one with a gene, and it's enough to perturb a pathway enough.
Yes,
to get some pleiotropia in terms of the disease.
It's a very good question. We have not done it. It's really easy to do in C. elegans. I mean, you can stack these things. CRISPR's working fine in C. elegans, so you can, these things grow in a week or whatever. So I mean, you can just knock these in. And we haven't done it yet. Chris, the C. elegans biologist, is anxious to get some combinations because he wants to do it. And you can also do up and down regulation. These are all RNA knockdowns, but you can also do overexpression. And again, you can do CRISPR. We're just really kind of doing a bulk analysis right now, but yeah. Multiple
disorders and bipolar. Sort of found it by imputing stuff from using the NIH epigenome roadmap, kind of approach. And so I was curious, and that was lithium response in bipolar. So I'm curious, have you investigated that in any more detail?
No, we haven't. I'm trying to remember if it was on the C. elegans list and whether
it
was
was.
A homolog.
It was.
It was, okay.
Also, PER1 was on there, which is a clock
that's
gene,
an interesting
right?
one, especially for bipolar. Right. Yeah, that's a phasic gene. Yeah. That actually wasn't. So it's been implicated in bipolar. It actually was a mutation in one of the schizophrenia, de novo mutation in one of the schizophrenia trios. Yeah, that's one of the ones where you look at them like, wow, that's interesting. But yeah.
So I'm curious about some of your the simulations of the SNPs and the exons. Did you do any work looking at what happens if it's not 100% exonic and if you have half in non-coding regions?
Yeah. I mean, it shifts it roughly double if
Cool.
it's half ascertained. I pulled that slide out just because I was running a little long, but it shifts it about. Yeah, it kind of doubles it. So you're looking at 1,000 genes. You're looking at sort of 20% power at like half a million.
So one of the big problems in psychiatric studies is the occurrence of multiple diagnoses in the same kindred, or same family. And you had examples of that. And you sort of have to deal with this. You have monopolar, bipolar, you have schizophrenias. We were talking about this, has to be described in the plural. You mentioned heterogeneity as number one. Then you have autism spectrum disorder, and all these sort of miscellaneous heterogeneous categories in the same families. It's a formidable challenge to sort this out. What is the strategy?
Well, yeah. As I was meeting people today, I was telling them I'm becoming more and more interested in probabilistic events. In other words, with a given genotype, you have a certain probability of being healthy or a certain probability. And there's epistatic interactions. There's probably random things going on. I mean, the real answer is we really don't know. How we're dealing with it is we're trying to focus. I mean, that is an advantage of working with families in that we, especially families that have been diagnosed by the same clinician, because there's at least probably some consistency, and there's some genetic consistency there too that is hard to recover in a big case control study like our bipolar exome study. You know, if I knew how to do it, we would have done. I mean, you know, I explained what we're trying to do. We're basically trying to, you know, look at families, get some candidates with a pretty wide net, and then zoom in on some larger samples, and then look at the function. But you know, we don't have an answer. You know, to be honest. Are you aware of the future clinical phenotype? Yeah, I mean, I've been heartened by the progress with trios in autism. I think that's really, you know, in the last 3 years, there's been a number of groups, and we've been a little bit involved in it. But I think it's really changed our understanding of the genes involved in autism by looking at large numbers of trios. That's starting to happen in schizophrenia, although there have been some large trio studies in schizophrenia that have not found a lot, although they are finding, you know, again, some hints and some networks that may be involved in that. So I think, I think that's one reason why we're kind of migrating towards functional assays as well as large-scale targeted assays, because I think we are starting to get some hints from these various studies. Thank you.